So starting with the PCR cycle, you start with a molecule of fructose creatine and a ADP molecule. This yields an ATP and a creatine. This is for your 1 to 5 seconds extreme burst of energy. Then you move over to your glycolysis. Glycolysis takes glucose from carbohydrates. So you start with the 6 carbons chain, and the 6 carbons run into hexokinase, which adds a phosphorus to the end of to the 6th carbon in the chain. This process takes one ATP, turns it to an ADP. This new chain is called G6P. The G6P then runs into the next enzyme, which is called PG isomerase. The PG isomerase transforms the chain into F6P. Then the F6P chain runs into the enzyme PFK. The PFK adds another phosphorus to the end of the chain. This process also takes one ATP, turns it to an ADP, and this new chain is called F16BP, which stands for fructo-1,6 biphosphate. This F16BP then runs into the 4th enzyme, which is called atolase. Atolase splits the F16BP into 2 parts, a DHAP molecule and a a DHAP chain and a G3P chain. The DHAP chain transforms into a second G3P. So then we have 2 G3P molecules. Through this process, the G3P chains take 1 phosphorus each from the outside environment, and then break off both of those phosphates, and it yields 4 ATPs, which is a net of 2 ATP because there's two of them originally used in the beginning of the process. So after that, you are left over with 2 chains of pyruvate. The pyruvate then goes into turns into acetyl-CoA because it loses a carbon dioxide molecule. This acetyl-CoA is a key for the Krebs cycle. So with one glycolysis cycle, you have 2 keys, as seen here, 2 keys for Krebs cycle. So each key turns it 1 time. The acetyl-CoA runs into citric acid, which binds into 6 more carbons. Then it runs through the Krebs cycle, producing 3 total NADHs and 1 FADH. It also creates a GTT, which then can be turned into an ATP. So the NADHs, all of those run into the electron transport chain cycle. So they run into the beginning of the cycle itself. They go to the first terminal that they can reach. So the terminal accepts the NADH, and this electron pulls the hydrogens off the NADH and pushes them to the inner membrane of the mitochondria. Then the electron moves down to the second terminal. After it does this, the NAD is released from the first terminal. Does the same thing, pushes 4 more hydrogens into the inner membrane of the mitochondria, moves back down to the third terminal. This time, since it's weaker, it only moves 2 hydrogens up. The FADH, on the other hand, comes into the second terminal, never the first one, only to the second one, which then this electron pushes up 4 hydrogens. Then the electron moves down, kicks out an FAD. Then in the third one, since it is weaker, it only moves up 2 hydrogens. So from there, the electrons have nowhere else to go, and to keep this process moving, two oxygens come in, they pick up the electrons, as well as the extra hydrogens that I'll get into in a second, and form metabolic water. So all of the hydrogens that were pushed into the inner membrane then come to this part of the ETC. So it takes 4 hydrogens to push through this crank, which has a inorganic phosphate attached to it. So for every, it takes 4 turns of this little crank here, and each turn is produced by 1 hydrogen. So with the 4 turns, it takes one of the ADPs that is on this side of it, turns it into an ATP, and then the hydrogens that are left over are then brought into the metabolic water. So this then results in, with 1 spin of the Krebs cycle, creates 3 NADHs, which is 10 total, which is 10 hydrogens each, and then 1 FADH, which is 6 hydrogens each. Then multiply that by 2, because there is 2 terms of the Krebs cycle per 1 run of the glycolysis, which then equals 32 hydrogens, which from this process equals 8 ATP. So from the entire process, starting with glycolysis to Krebs cycle to ETC, produces a net total of 2 plus 1 plus 8, which is 11 ATPs.